All right, thank you everybody for joining us today. My name is Ashley Grossman. I'm the membership coordinator for the Society for Simulation in Healthcare. We're presenting a webinar today hosted by the Healthcare Systems Modeling and Simulation Affinity Group. Just a couple brief, I put in the chat box to please mute your audio if you're not speaking to help eliminate any background noise during the presentation. We're also recording this presentation and I'll be uploading it onto our SimConnect community for this affinity group here sometime in the next couple of days. So keep an eye out for an announcement about that. Next, I'd like to turn it over to Dr. Yue Dong, who will be introducing the presenters we have lined up for today. Thank you.
Hello, good afternoon, everybody. So this is Yue Dong from Mayo Clinic, Rochester. Welcome to our monthly webinar for the affinity group for this healthcare system modeling simulation. So this is affinity group from the Society Simulation for Healthcare. We have been formed last 4 years. We have currently 2,000 membership in our group. So today we are fortunate to have Dr. Mary Kay Smith from she's the director of the Learning and Assessment Center from the Michigan State University. And she's also in the chair for the accreditation committee for the society. I've known her from the work in the society many years. I'm very glad to have her join us. Also, we have another professor, Dr. Laura Bix from the MSU School of Packaging. So this is very interesting. We bring a colleague from another industry to share their expertise with us. And Dr. Smith and Dr. Bix have formed an unusual collaboration research endeavor. Let's try to objectively investigate the interaction between the people and the packaging using healthcare simulation. So ultimate goal for their work is try to employ insight gained to improve healthcare outcome. So before we turn over to these to Dr. Smith and Dr. Bix, I also have an announcement I want to share with the audience. In 2018, January 12th and 13th, we will have a simulation forum on modeling simulation and for simulation of application beyond training education. So this is a great opportunity for people interested in using simulation to apply innovative work in healthcare and settings. In order to prepare this meeting on May 3rd, next slide please, on May 3rd, at 2:30 Eastern Daylight Time, we will have a Twitter chat. Invite all of you to participate to get some ideas to us how we want to organize this meeting. I hope all you can join us. So without further ado, I'm going to turn over mic to Dr. Smith. Please go ahead, Mary Kay. Thank you.
Okay, thank you so much for joining us today. We're honored to share what we've learned from each other and working with each other in our journey. So we'll begin by looking at our ultimate goal, which is to ignite innovative research collaborations using healthcare simulation to improve healthcare delivery and ultimately health outcomes. So I think you'll find that our goals and our underlying objectives are actually the same regardless of our approach. Today we're going to examine methods by which non-traditional collaborative entities can utilize simulation to the benefit of healthcare research, identify benefits of non-traditional research collaborations within the context of healthcare simulation, explore some exemplars of non-traditional research initiatives between the Healthcare Simulation Center here at Michigan State and the School of Packaging at Michigan State University. And then we'd like everyone attending to recognize one potential research collaborative opportunity utilizing simulation within your own organization. Our agenda, we're going to begin by sharing a little bit of our story, like what brought us here today. We're going to share one of our major projects that we work together, collaborate with, which is the healthcare packaging immersion experience. We're going to present some benefits and justifications of non-traditional collaborations, and then we're going to provide some exemplars of research leveraging various aspects of simulation. So now, as we look at our story, it's really been amazing as we've evolved here at Michigan State over the years. We were, like many, in our silos, doing great work, but yet not necessarily doing great work together. So probably over a decade ago, Laura, Dr. Biggs and I became acquainted with each other through a mutual colleague. I was working in the College of Nursing at that time and was invited to share perspectives regarding aseptic technique with the School of Packaging students who were focused on healthcare packaging and medical device packaging. So that was when my eyes were open to this whole new concept of working with a non-traditional group of individuals. I never would have dreamt 30 years from ago when I entered the workforce that there would be this common denominator. So as time went on, we gradually grew and learned from each other. I learned a new language, I believe, as Laura has shared as well. We've learned a lot with and from each other, which I think if we think about the World Health Organization and their definition of interdisciplinary care, I would say that this is certainly an exemplar of that and a different one of that. So a few things that that kind of prompted or provides the underpinning of our work together is, adverse events. The fact that many people are injured every year due to medication errors. Of course, the Institute of Medicine reports, multiple reports now over the years, have all pointed towards the need for better interprofessional care. But what we often think about is the nurse-physician interface and not necessarily all of the other partners in healthcare that we have. A few other areas that we might want to consider as we think about the research is challenges that packaging can impact on healthcare. One being nosocomial infections, looking at the incidence of those and how those can occur, and then taking that a step further and think about how packaging can impact that. How we, as end users in healthcare, utilize packages and how the design of those packages has a direct impact on our ability to maintain sterile technique or not. So thinking of the failure to present aseptically may be blamed on the provider when in fact the package may not have been designed with the end user in mind. So another area that we might think about is labeling. Many of us can relate to this as we practiced or practice at the bedside and direct patient care, how there are some times where labeling is very similar and can predispose or increase the risk of causing an error. And here's another area which I had no idea, to be perfectly honest, that this occurs until Dr. Bix and I began working together and actually work with some of our colleagues here at Michigan State with regard to counterfeit drugs in devices. This is becoming more and more prevalent and a problem as well. So how can the packaging industry impact the end user's ability to identify authentic versus counterfeit medications or devices. So we began to move from our silos to bridges together here at Michigan State as we realized we had so much in common. And I'm going to show you a video of a physician that engaged in a simulation that we designed it was an emergency room simulation, very chaotic, multiple packages were opened, and we debriefed with the healthcare providers that were facilitated and engaged in the simulation and the packaging engineers that attended a conference. And it was the comments from one of the physicians that participated in the simulation that really brought home what we were thinking.
Regarding the package in front of you, in real life, if you would have experienced that, would you have reported that or given that feedback to somebody to get back to the manufacturer, or do you just chalk it up as a learning experience and move on?
To me, it's a learning experience because it's. I would think of it as my fault, and I would yell at the nurse, maybe like, "Why is this so hard to open? Well, how do you open this?" I mean, most doctors count on the nurses to know how to do opening stuff and things like that, and no, I wouldn't go beyond that room. Like I wouldn't say, gee, we need an in-service on how to open these copper tubes. No, it'd be a learning experience for me, and yeah.
Then when Dr. Jekyll is done yelling at the nurse and drops it on the floor, he would not be allowed to reuse that. I know that he said it's okay, we can use it, but no.
This is kind of nice to have this forum because we get to hear from you guys and you get to hear from us. And you said when you walked in here that one of the reasons you were here is because you were traveling all over the place and you're finding different ways of doing things.
Yeah.
What do you think your avenue is to get back to us? I know packaging is at the very, very bottom of all the stuff that you guys have to worry about, but we don't know out there that you are finding these problems, or at least I don't know. What do you think your avenue is to get back to us this information that we need to give you what you need?
Yeah, honestly, until yesterday, I didn't realize you people even exist. I mean, I'm not saying that to be negative. It's just that a whole world. I mean, I thought this came like from a black box. I didn't realize that there are. I mean, and sometimes prior to yesterday, I didn't realize people actually give thought to this. I mean, sometimes I'll do stuff and I'll say this is not a medical person behind this, but you guys are like a black box. I think to most docs in the front line, we see drug reps a lot. We see a lot of pharmaceutical reps, and I think probably surgeons see like product reps. But I would be shocked if any ER doc, other than maybe a department manager, has met with a packaging person or people who think the way you guys think. Like the doc.
Okay, so we'll cut it off right there. But how many of you, without answering in the comments, can relate to that? Being an end user and not having any idea that anything went into the design of a particular package or medication or device, the list goes on. So this was not so much an eye-opener for us, but really solidified our need to work together, and yet brought a question up as to why haven't we been doing this for years? Why is it the 21st century and we're just beginning to interface? But not being able to change the past, we are looking forward to what we can do now and in the future. So here is a comment made after that particular conference as well, in which this work really resulted in many lively conversations, many more thoughtful packaging designs, positive impact on the industry, and ultimately, again, how this all translates to better, safer patient care, again, our common denominator. So then this really, as I said, solidified our thoughts, and we decided to move forward, do more, and we branched out into what we see as 3 different areas, one being education, outreach, and research, being our triangulation of work together. With regard to education and application, we facilitate work with the students. We have undergraduate packaging students that are trained in aseptic techniques so that they can understand and hopefully are more cognizant as they design. They gown, they glove, they present sterile packages on a field. We simulate stressful environments so that more understanding can be made to human factors, etc., which then impact labeling issues and presentation. And then there's a number of students that observe nurses actually in training with regard to aseptic technique. Another area of education that we've been focusing on is physicians, nurses, surgical techs, probably being our primary participants, and they will engage in simulations here at the center, provide their feedback, and again, another forum in which packaging designers, engineers, researchers can interface and identify issues with various packages and medical devices. And then with regard to outreach, our primary outreach activity is what we refer to as HICPI, and this is an acronym for Healthcare Packaging Immersion Experience. This is a 4-week conference for the industry. So in the previous video, that simulation had occurred during that particular conference. We have nurses, healthcare professionals, packaging students that all contribute to this 2-day event. So everyone from various perspectives contribute to this, and it's well received. We've now facilitated, I believe, what is it, 4 or 5? 4 every other year, and it's very well attended. We also have, as I mentioned, undergraduate opportunities to liaise with speakers and experts, and it provides a platform then for further work and research. So moving on to what brings us here today, we're going to take a deeper dive into, again, the third area of our triangulation, which is the research initiative. So I'm going to turn it over to Dr. Laura Bix to share with us.
Yep, so I'm just going to highlight a few of the types of studies that can be done with simulation of various types. One great thing about working with Dr. Smith is I have a better understanding of the breadth of tools that are involved in simulation, from partial task trainers to gaming strategies. We're trying to implement sort of objective measures into what has traditionally been an artful sort of endeavor to the benefit of the provider and ultimately, hopefully, the patient. So in terms of sort of how we view this is kind of a visual abstraction of how my research team gets things done. I lead a group called the Packaging Hub. That stands for Healthcare, Universal Design, and Biomechanics. And in a nutshell, what we try to do is employ a user-centered approach to package design that is informed in a quantitative way with the goal of improving health outcomes. And this is done through an engaged process across the mission that Mary Kay has already shared with you, in an attempt to create a cohort of scientists and industry professionals that embrace more of a user-centered approach, that enhance accessibility in packaging and change the industry's approach to design in order to improve or move the needle on several of the problems that she's already shared with you. Adverse events, nosocomial infections, unintentional exposure of children to household chemicals and drugs, and reduced rates of healthcare-acquired infections. Traditionally, in the packaging industry, we tend to be very product-centered in our approach to design. That is that the properties of the product determine sort of the design aspects, that the product has to be sterile. The product has to maintain sterility throughout distribution and handling. And traditionally, that's come almost exclusively on that side of the sort of slide. More and more, we're embracing a user-centered approach to design, where the user's functional capabilities and the user's needs need to be at least part of the equation in the decisions that we make. And so we're trying to incorporate that into our curriculum. We're trying to gain insights from the work that Dr. Smith does in insightful ways that our students will then go off and make better products. So just a little bit of background that I think you need to really kind of fully appreciate some of the research that we're doing. In the 1970s, the perceptual psychologist James Gibson coined the term affordances. And affordances basically mean that there are objects in the environment afford an actor actionable possibilities. So in this example, the stump can serve a lot of different actionable affordances, if you will. It can serve as a resting place. It can serve as a watchtower. It can serve as a table to eat. Or for some people, not myself, it can serve as an exercise object. So the actionable possibilities under James Gibson's construct are independent of the observer's ability to recognize them. That is, that stump serves all of those purposes regardless of whether or not I realize that it can be used as such. But a few years later, in the 1980s, cognitive psychologist Donald Norman sort of expanded the concept and brought it more into the world of industrial design by saying that affordances really didn't exist unless the actor perceived them. That is, that they had to convey their meaning through sort of a design factor to the actor in order to really be effective. And so it took it out of the world of psychology and put it more into the world of design, saying that your design is sort of flawed if it does not communicate with the user the actionable possibilities or the intended sort of tasks that you're trying to convey. And Winder sort of polished this idea even further by saying that communication was a combination of the meaning that you were trying to convey, that is, the task that needed to be accomplished, but also the strength of the signal. And what she said is that some signals are so strong that they cross species. So like a bowl of water to a thirsty human or a thirsty puppy is so strong in terms of its signal strength that both of those actors, even though they're different species, even though maybe they're different developmental ages, can understand that they can drink from it. And so one of the things, and I give you that as a background to some of the research that we're doing and sort of the construct that grounds some of that research. So one of the projects that I want to highlight today, and I have three that I'm highlighting, is packaging as a potential route for hospital-acquired or healthcare-acquired or nosocomial infections. And one of the things that we got very interested in was the relationship between packaging design, the biomechanical approach to sterile procedure or aseptic technique, and contact with non-sterile surfaces. So Mary Kay has already kind of highlighted this for you, but a healthcare-acquired infection is defined by the Food and Drug Amendments Act of 2007 as an infection that is acquired while an individual is a patient at a hospital that was neither present nor incubating in the patient prior to receiving services in the hospital. And so we know that these are a significant problem. In fact, it's been estimated that as many as 1 in 25 patients in the U.S. has at least one HAI on any given day. Other researchers have suggested that there are 2 routes of transfer for these infections. One is a direct contact transfer, that is person to person, and the second potential route is an indirect contact transfer, that is through a contaminated intermediate object or a secondary person, if you will. And so as we started looking at the literature, being in packaging, we started to get interested in packaging as a potential route of indirect contact transfer. The package, you all as an audience know this, the outside of the package is not sterile, and so only the circulating personnel or the anesthetists can interact with the outside. But the inside and its contents very frequently, not always, but very frequently are sterile, and only those in the scrub rolls can interact with the inside. And so aseptic technique is defined by the Association of Surgical Technologists as anything used to achieve asepsis, which is an absence of microbes. They practice sterile technique or action taken to prevent microbial contamination of the sterile field. When we started kind of looking into packaging as an indirect route of contamination, we really found a dearth of literature in terms of indirect routes of contamination. Most of what's been done has looked at or focused on direct contamination, direct routes of contamination, focusing on things like hand hygiene practice, checklists for appropriate gowning and gloving, and the like. What we did find in this space was pretty limited. There were 2 studies that looked at double barrier, and did double barrier result in excess handling? And there were some things on exposure time and storage conditions, and a little bit on the hand contamination of the circulators and how that might play a role. But most of it was really kind of focused sort of squarely on blaming the healthcare provider, quite honestly, and not at how do the design of things that the healthcare provider has to use to accomplish their job impact their ability to accomplish their job. And so one of the things that we used was we adapted a method that we found from Crick, who was a doctor, I believe, in Australia, that was written in 2008. And what it does is it uses an analyte called a glitterbug or glow germ. This is commonly used in hand hygiene and sort of contamination studies. This analyte is, in the case what we use, it's a lotion that you rub into the hands of the person, and it transfers very readily to other surfaces. We rolled it on the outside of the package and on the backs of the hands of the provider. It is invisible under standard lighting conditions, but it phosphoresces under UV. So there's no signal to the person, sort of feedback loop regarding, oh, that came in contact with the non-sterile surface. So what we do is we have different treatments, and then we have the providers make the transfers, and we observe the device for contact with a non-sterile surface during the transfer. And then we get a binary result, contaminated, yes, no, that can be analyzed through a variety of statistical techniques. The work initially was published by Trier, who looked at the effect of pouch size on rates of contamination during simulated presentation to a sterile field. And actually you can see that he found a significant effect of size, whereby the large trials were contaminated at a rate of almost 15% of trials were found to come in contact with a non-sterile surface. Now that doesn't mean that that's going to result in that it's going to result in either contamination or a healthcare acquired infection. What we can say is it came in contact with the non-sterile surface during the transfer process, and that the larger pouches were significantly more highly contaminated than the smaller pouches, which were only about 6%. When we started sort of analyzing, we also looked at the number of repositionings that the providers used when they opened the pouches. And the large pouches had a significantly higher number of repositionings than did the small pouches. And so we work with a biomechanist, Dr. Tammy Bush here on campus. She started to see her profession in this result, saying it was the increased handling that resulted in these higher rates of contact with non-sterile surfaces. I started seeing my profession saying it was the large corners which had a tendency to roll in, which resulted in the higher rate of contaminations. We went back to our video and analyzed post hoc and found that about 50% of the trials we could verify that the materials tended to curl in. We also started to look at where did the providers begin their sort of motion or their opening. And there were fewer providers that began from the middle with regard to the large pouch. And that gets back to the affordance theory that I discussed before. Specifically, in the large pouch, we have these large corners here. And what those large corners do is they signal, they afford the strongest grip that you have. You have 3 different types of grip. You have a tip pinch, which is sort of the tip of your finger to your thumb. You have a key grip, which is your strongest grip, and that's like you would grip an old-fashioned automotive key. A lot of cars don't have them anymore. But what that large corner does is it lets you get this much more powerful grip. And so we saw that a lot of people were gripping in these, in the larger pouches, were gripping out here. Well, that brings to question another interesting research question. This is the outline of a tray, and if you think of peeling the lid of this tray from this point forward, as you peel, you get a larger and larger line of the peel, W sub 1 here. The larger that line we've shown on our Instron machine, the higher the force required to peel. And so one hypothesis that we've had and we are pursuing right now is that these large drops in force, these large differentials, when you go from a large line to a smaller line, they cause jerking actions. So if you've ever opened like a juice cup and you've opened the circle and gotten to a long line and then 2 little lines, that's where we're hypothesizing it's likely to have a jerking motion.
Hi.
So when we give the providers this large area to grip, what we do is we get to a situation where they go against a long peel line, where if they had peeled the way it was intended, straight down, you have a much more even sort of length of peel, which would result in a more even and flat distribution of forces. But we've created a situation where we've sort of encouraged them to grip in this larger area because they have a stronger grip, which creates a situation, potentially, we hypothesize, that as they go from this long line of peel to this small line of peel, we end up with this jerking motion, which is more likely to result in contamination or contact with a non-sterile surface. One of the things that we're studying right now is as we change different seal geometries, can we characterize the forces that result, and can we also use the technique that Trier used in his previous master's work to show that they result in different rates of contamination or different rates of contact with non-sterile surface. And so this work is being done by Paula Perez. It's founded by the Blue Cross Blue Shield of Michigan Foundation. And she is just today finishing up her final data collection. She is looking at how peel geometry and peel path impact contact with non-sterile surfaces, characterizing the jerking motion. She also have treatments that curl in and curl out. The hypothesis being that when the material tends to curl in, it's much more difficult to aseptically present it. I never worry that my banana is going to be hit by the outside of my banana peel, for instance, yet we create these packages that healthcare providers have to fight with. Another study that we've done that employs gaming to look was also funded by the Blue Cross Blue Shield of Michigan Foundation, looking at forced choice tasks. Specifically, we heard from providers that labeling on medical devices, which is very unregulated and not standardized, were difficult to read. Specifically, they indicated to us in focus groups that non-critical information interfered with the information that they needed, and that they had 4 pieces of critical information that they needed. That was the expiration dating, the latex status, the sterility status, and the product name. Some of the recommendations that they made was that grouping and highlighting of information, standardized locations, bigger text, brighter colors would make things easier for them to read. And so we did a benchmarking study on indwelling urinary catheters. We had 6 companies and 20 products, and we characterized in quadrants those 4 pieces of information that they said were important to them. That is latex status, sterility status, product name, and expiration date, to see were they indeed segregated or separated the way that the focus groups had indicated. 40% of the products that we used had 2 locations. 60% that we used had that critical information in 3 locations, and none of the 20 catheters that we studied, pulling them randomly from the market, had all 4 pieces in the same quadrant. And so we said, wow, this is something we need to objectively characterize. So we created mock labels based on the commercial labels that we were seeing. These are really commercial labels that we sort of created a mock brand to protect the manufacturers. But these are 2 labels that we used in the study. And then we looked at the impact of color, grouping, boxing, and symbol usage on time to correct identification of a product and the proportion of correct identifications of products. We crossed all of these conditions and very carefully controlled things experimentally, and we randomized. We would yoke the labels, and I'll show you on the next screen so that's more sort of apparent how it worked. But we randomized the position of the information so that people didn't learn to look for particular pieces. So this is what a trial looked like on the computer screen. This is a label that has color, symbol, grouping of the critical information, and boxing of the critical information. The only difference between these 2 labels is that one indicates that it's latex-free, the other indicates that it contains latex. So for each trial, we would ask them to select as quickly as possible along a given dimension. It might be what product has latex, what product is expired, which product is sterile. And both of the labels were at the same level of treatment. And so the proportion of correctly identified labels and the time to correct selections served as our dependent variables. In terms of the binary response, which is the proportion of those correctly chosen, we found that boxing and symbol use both had a significant effect. However, boxing went in the opposite direction that we expected it to. So when the critical information was boxed, people were actually less likely to choose the correct product than when it was not boxed. When the symbol was present, it went in the direction that we expected. That is, that people were more likely to choose correctly. In terms of time to correct selection, grouping, symbol use, and color. And color was done in a very simple way. It was binary, red indicating sort of caution. So if it contained latex, if it was not sterile, it was in red. If it was latex free, or if it was sterile, it was presented in green. So I know color can be kind of controversial and can go in different directions. But we found that grouping, symbol usage, and color all enhanced time to correct selection. And so these are some of the sort of comparisons. You can see the difference between the symbol being present and absent in terms of time, the information being grouped and not grouped, the information being color-coded and not color-coded. Now, when we compared the optimal trials, that is grouped information with symbol present and color-coded, to the commercial labels that we had designed, you can see that we had significant difference in the performance rate along the correct selection, whereby the optimal trials were chosen correctly about 97% of the time, as compared with commercial label A, chosen about 92% of the time, and commercial label B about 89, almost 90% of the time. And then in those trials that were chosen correctly, optimal label A, the optimal trials were selected significantly more quickly than either of the commercial labels. And so we take this as objective evidence that we can really make improvements in the way that we design things for healthcare providers. So the final study is an evolving study. Mary Kay is on the committee that is working on this, and that's that context significantly influences the affordance behaviors. That is, that a pre-hospital environment and the behaviors that are exhibited in a pre-hospital environment are going to be significantly different than a perioperative environment. Specifically, we have done a survey through the National Registry of Emergency Medical Technicians to ask about the opening practices of paramedics and EMTs in those environments. And we found pretty significant rates that one-handed use is kind of the expectation as compared to two-handed use for these packages, that they're very frequently using trauma shears to open sterile packaging, which could present a problem. For instance, if they're using those same shears to cut bloody, maybe dirty clothing off of a patient, and then the next patient comes in and they cut a sterile package open with that. And that teeth are frequently implicated. And so what we're thinking is that when my profession, that is packaging professionals, do bother with regard to garnering user insights, they're tending to go to perioperative environments, environments where you have planned roles, where things are more calm, where you have two hands. And then after we've developed it, we throw those same products into the battlefield, onto the life flight, into the back of the ambulance, and we expect the affordance behaviors to be the same. That's not the case at all. So one of the things that we did this summer and this past summer and winter was we partnered with Ohio State University, the Wexner Medical Center, and the Lansing Community College, and we commandeered a couple of their ambulances. Here in the mid-Michigan area, we outfitted the ambulance with triaxial accelerometers so we could collect vibration data from in and around the city, going over the potholes and through the chuck holes. And then in Columbus, Ohio, we rented a test track in East Liberty, Ohio, that's commonly used by Honda, and we put an ambulance to its paces. We sped it around the test track, we weaved in and out, we drove over curbs, we drove over potholes and chuck holes, and did a lot of illegal things that they wouldn't let us do on a non-closed course. The intention is that we're going to take that vibration data and run a vibration table that's a 6-axis degree of freedom vibration table at an automotive supplier in Detroit. and actually run simulations with motion, light, and sound incorporated. And we'll ideate around how we can design things that will work better for those environments. So I think the takeaway really is that the complex problems of healthcare require thoughtful, data-driven inquiry that utilize varied perspectives of expertise. There is not going to be a singular solution for adverse events, for hospital or healthcare-acquired infections, but novel thinking has the potential for impact. And I think that is where, you know, really partnering in uncommon and unusual ways has the potential to move the needle on these issues. In addition, our students have gained understanding. They have a platform in terms of the HCPIE event to present their research. The industry has an opportunity to come and see what our findings are, to provide their input, as do the healthcare providers, regarding, oh, no, you're recruiting the wrong type of person for that. A paramedic is not an EMT. I didn't even know that until I started working with Mary Kay. I had no idea. So it really, in terms of the benefit, it's been beneficial in many, many different ways. The money that we generate from the conference funds the research, which is then presented on the stage, provides equipment for the LAC, and gives us the opportunity to develop pilot data that can then be spun to organizations like Blue Cross and NIH. So looking ahead, we are continuing the collaboration and looking for novel and new ways to learn about healthcare and improve what's happening. So with that, any questions? Is anyone still there?
Yeah, so please feel free to ask questions for Dr. Biggs and Dr. Smith. So Mary Kay, may I have a question first? So traditionally, we are healthcare professional. We are on our campus. I'm not sure the layout for your school. So how you first initiate this kind of a discussion? How we really start this collaboration? Because I see that's a lot of opportunity for collaboration across the traditional boundaries. Can you just tell me a little bit more about the story when you first met Dr. Biggs?
Absolutely. At the time, I was working as faculty in the College of Nursing, and some of it was just, to be honest, fate or chance. One of the doctoral students in the School of Packaging, who actually it was one of Laura's advisees, approached me to present to the packaging students with regard to aseptic technique. What is it? Why does it matter? Why should they be concerned with the design of packages and the end user? I brought some exemplars of the good, the bad, the ugly, as we all can probably relate to. And then it kind of grew from there. I really enjoyed the opportunity to interface with a different discipline, a different group of colleagues here on campus. And then we started meeting more regularly to brainstorm and think about various projects that we might be involved with. And then that doctoral student went on to work in industry when she graduated and came back, and it touched on Laura and myself to let's think about industry and how that relates, or how we bring that into the whole spectrum. And so and then HICPI was born, and then various research projects, and it's kind of grown from there. And as you can see, we've used a variety of different simulation approaches or methods, low fidelity, high fidelity, computer or gaming, just a real, we've become friends.
Yeah.
so we, it's, in fact, I was just before the presentation, we need to do something new. Let's think of something, we kind of push each other to do something new and different.
So very good. Thank you so much. This is really something which for all our group really try to bring the as a bridge between those two community, engineering community or healthcare professionals, how to see the value simulation to be added to the healthcare in general. And the other thing, this is not the traditional simulation center activity, training, education, or teamwork. How you see this kind of work in the future, growth of simulation community in general for the society members, other things to explore other opportunities.
I would hope that it would increase. And to be honest, when I first presented this to the deans, we serve the 4 healthcare professional colleges here at Michigan State, our simulation center, and the deans were, well, Mary Kay, go for it. Whatever you think. So I had a lot of, administrative, well, a little bit apprehensive. Are you spending your time wisely? After they saw the results of the research that was generated from all of these different initiatives, it was very interesting to hear the deans say a few years later that, wow, we thought you were crazy, a couple of years ago, but, who would have known, that this would grow the way it has. And I guess, I would challenge anyone working in simulation to think inside the box, think about opportunities within your organization, collaborative opportunities within organization. We don't necessarily have to go very far to identify opportunities like this.
No, thank you so much, Mary Kay. Also, I have a quick question for Dr. Bix. Thank you for joining us. Today, share your expertise with us. We are looking forward to more opportunity to collaborate with you and other colleagues. So, can you tell us, how we can do better as healthcare professionals, or more openly collaborate with you guys, because not traditional, we are different industries, there's there's a silos, have been in place. I think we try to learn, at least from our perspective, we try to learn from you guys, how we can helping patients better care, better quality of life, how we can do better from your standpoint, so engineering professionals.
Well, I think that, really just being open-minded and recognizing that the silos exist and that there are potential. And when I look at the literature, and I look from a very different perspective, it seems to me like healthcare providers get blamed a lot. If you look at the way that we're trying to tackle healthcare-acquired infections, it's smarter use of antimicrobials, it's better hand hygiene techniques, checklists for people that need to, and so I think we need to start thinking about designing the system for better health outcomes, and that means that my people are culpable just as much as that poor provider that's trying to do their best to deliver, care under maybe adverse conditions. I'm hopeful that as things like the universal or the unique device identifier and the electronic health record come online, that we'll be less focused on procedure by procedure by procedure, and we'll be more sort of comprehensively focused on health outcomes. And as the reimbursement models start to move toward reimbursement for health outcomes as compared to procedure, I hope that it'll force us all to look more systemically at things.
I have a question. Have you been able to translate any of this into what could be the potential financial return on the design changes?
No. The challenge that I have that I alluded to before is you can't prove, that an infection will occur unless we do some type of animal studies. And so that's really the missing link. I can show that it came in contact with the non-sterile surface, and I can show the factors that are more likely to result in contact with the non-sterile surface. But to take that all the way out to an infection, and then it's going to be patient dependent, is difficult. But like in the case of the labeling, I can't prove that they would have chosen a latex containing product, but I can prove that one design is more likely to result in the correct choice. So I'm looking for partners that can help me make that argument better than I do.
I would think there'd be a way that you could do almost a before and after study of after a product design changed to see if that was the only thing that changed. Did it impact an infection rate?
Yeah. The problem is that my people, medical device world and the packaging world, that's a Pandora's box for them, right? Because you guys get blamed. You guys are the ones that get sued. So, when we go to NIH or somewhere like that, they'll say, well, why isn't the industry funding this type of work? And, well, the industry's not funding it because they don't necessarily want to know, right? So we've had some luck with the insurers, but if we can make the case to the NIH, I think we'd have better chance for federal funding.
I think also another caveat to that is access to real patients for the or by the industry, and this is where simulation can play an integral part too. Back in the day, they used to be able to take their devices and packages into or in situ, and providers would just utilize them. That's not tolerated anymore. It's not legal anymore. So again, simulation can fill that gap. As again, not directly answering your question, but came to mind as I was listening to Laura as well.
Dr. Fix, there's another dimension. Maybe you can help us to see this. I know in the business world, the time to market is critical, important metrics, for business success. I think for this case, for design, for development or testing, basically simulation afford a shorter time to market, because this is really a huge win, and for industries to provide value for the customer, for the patients, providers. Although they're not necessarily a better product, but they are faster to deliver better product to the market. I think we may see other angle, look at the ROI instead of just patient outcomes.
Mm-hmm. Well, I think part of the problem with the ROI is until the hospital starts purchasing based on this type of thing, it hasn't. I think a lot of times it's a piece part decision as opposed to an overall healthcare cost of care decision. Mm-hmm. And that's why we need these systems, the electronic health record, the unique device identifier, to tie everything together. Because I think right now a lot of the purchasing decisions are made on the lowest piece part as opposed to what results in fewer days in hospital.
Though I do have to also, I can't help but think of the companies that utilize simulation to test their product before it's rolled out into market, and the flaws that they identify in the products and the packaging. I can't help but think of the thousands of dollars that saves companies in with regard to forging ahead with a certain design and to have it fail.
Yeah, I think that's exactly right. Last week in the medical device conference in Minneapolis, hosted by the University of Minnesota, there are some officers from the FDA talk about this. They are really looking this those kind of testing, for providing regulatory evidence to supporting FDA approval. I think this is really on the agenda. Actually, they are talking about not just for this kind of simulation. They are talking more on the computer modeling simulation to testing devices or implants. I think this is on their radar because they want to do more and more in silicon clinical trials. In the future to accelerate this process. So is that what you heard, Laura? In the conference.
Yes.
The conference. Yeah. I think this is
and
coming.
There was a guidance document in February that specifically addressed packaging and usability.
Yep.
And so in the validation protocols, traditionally it's been very product focused, but they're starting to reference usability and utility issues with regard to the packaging in the federal guidances from CDRH. So I think that will push things along too.
Sure. So if you do mind, Dr. Bix, would you mind send me the link? When we have to post the video online, we will put that link into our internal SimConnect site, also the YouTube channel, so people interested in that topic can look at more information.
Fabulous.
Okay. So any questions from audience? So if no, so I think we can just.
It's dinner time.
Thank you, Dr. Bix, Dr. Smith, for your time to share your excited project with us, and we are grateful. So you'll be part of this efforts to bring simulation to the next level. So, and also thank you for all the participants. So we'll see you next time.
Thank you.
Okay, thank you.
Bye-bye.
Thank you. Bye-bye.